Hi, today we're going to be talking about DNA replication, protein synthesis, and mutations of DNA. So first we're going to start with DNA replication. So what DNA replication is, it's the process where a new DNA strand is created from an old one, which is shown in this picture. For DNA replication to occur, there are many enzymes that do different tasks that work together in this process of making a new strands of DNA. So first, we have an enzyme called helicase. So what helicase does is it splits the original DNA strand of DNA into two. So as you can see right here, it splits it into two. And it's important because it allows the other enzymes to do their jobs in the future. Next, you need something that could read the bases. It needs to read the bases so it can lay down new ones. What a base is, it's these tiny little ladder rungs, and these bases sort of make up our physical traits. So that's the job of something called DNA polymerase 3, which is this one right here. So it reads the bases of the DNA and lays down the corresponding base. So there are four bases: adenine, thymine, guanine, and cytosine. So these bases, when they're put in a certain order, it makes up our physical traits. So each base is matched to a certain base. Cytosine is matched to guanine, and adenine is matched to thymine. So what the DNA polymerase does is it reads one. So let's say it reads adenine, it'll lay down a new thymine. So, but the DNA polymerase is a little dumb and it doesn't really know which direction to go to lay down the new bases. So he needs the help of something called the primase. So what the primase is right here. So what the primase does is it sort of acts like a little guide and tells the DNA polymerase where to go. So it creates a negative charge. So it creates like a negative charge right here by making RNA, and it'll sort of attract the DNA polymers. So the DNA polymers reach from 5, which is right here, to 3. So it just follows that, and then it'll create new bases. On one side, it just has to create one negative charge, like right there. But then on this side, since it's from 5 to 3, it has to create multiple ones. So it create one here, it'll create one here, it'll create one here. So it makes sure like the polymers keeps going in that direction. Since it normally doesn't go that direction, instead it goes this direction normally. So it has to make multiple ones. So now that we make the new bases, we need to find a way to seal up these RNA things right here that the primase made. So now that's the job of the ligase. So what the ligase does is the ligase, it sort of like acts like a glue. So it seals up the spots and it combines these things, which are called Okazaki fragments. And so it pretty much glues the DNA polymers together and it creates a new strand. So next, we're going to be moving on to protein synthesis. Now, essentially, what protein synthesis is, it's going from DNA to proteins. So this is just a basic overview of what happens during protein synthesis. So we're going to be focusing on how it happens in a cell with a nucleus, which is called a eukaryotic cell. So a DNA is a double helix, which is sort of like this. And then the nucleus has tiny little pores right there. But this double helix isn't big enough to fit through that. So it needs to make it into something called mRNA, which can fit through it. So I'll get to sort of like how DNA turns to mRNA like more specifically in the next picture. But for now, we're just going to say DNA turns to mRNA so it could escape the nucleus and goes to the ribosome, which is right here. So the ribosome, it pretty much what it does is it creates proteins. So the ribosomes read the proteins by groups of 3. These groups of 3 are called codons, and each group of 3 codes for a certain amino acid. So the mRNA will go through the ribosome and it will create these amino acids, which will eventually connect into the strand, which turns into a protein, which codes for a specific trait. Alright, so now we'll talk about how the DNA turns to mRNA in something called transcription. Alright, so in transcription, the DNA strand needs to be transferred to mRNA so that it can fit through the pores from the last picture right here. And so what happens is there's something called the RNA polymers, sort of just like the DNA polymers, and it comes along and reads these bases, and then it lays down the corresponding bases as an mRNA. But there's something different about the RNA that is created. Instead of matching up diamine, which is a T, with adenine right here, it replaces the diamine with something called uracil. So which is a U. So instead of A and T, it's A and U. So adenine with uracil. So that's called transcription. Now we want to turn the messenger RNA into a protein. So that's called translation. So I'm gonna describe translation. So after we turn the DNA into mRNA, that's when we start going to translation. So that happens in the ribosome, like I said earlier, in groups of 3 called codons, and they're read by something called transfer RNA or tRNA. And then after the tRNA reads the codons, which is a group of 3, it creates an amino acid based on what it reads. So in this 3, UGG, it creates something amino acid TRP. So same thing with UUU, it creates PHE. So it does that with each one, and it combines it into a strand like this one, and there's a lot of them, and then that makes up a protein. So that's how DNA turns into a protein. So now we're going to move on to mutations. Alright, so there could be many causes of mutations, but simply what a mutation is, it's a change of genetic material. So there could be different causes for mutations. When a mutation occurs, it could change the DNA strand and affect a person physically, like they could grow extra finger or something like that. But sometimes mutations have no effects at all. So a mutation could be an external cause, like radiation or like chemicals through food, or they could be internal. So one type of internal cause would be gene mutation. So there's about like 3 types of gene mutations. There's one if a base is deleted. So if you see here, in the original one, it's ACG. They removed the A out of it. So now all of these amino acids are read wrong. So when that happens, you get the wrong protein, and then the wrong protein leads to sort of like a mutation of a physical trait. So the wrong physical trait. And that's just another example. If they take out a whole codon, as you can see here, this becomes a lot shorter, and you're missing one. So that could also create a wrong physical trait. Another one could be insertion. So here they added an A, and they just created like a whole different new chain of amino acids, which again cause the wrong physical trait. There could also be a possibility where a wrong base is matched during DNA replication. So if you remember how, as you say, adenine, we have adenine, it normally matches to diamine, right? But then let's say the DNA polymers, which if you haven't forgotten, it reads the strand and creates a new one. So let's just say it makes an accident, and instead of diamine, it matches it with guanine. So that could cause the tRNA to read it wrong and again cause a wrong physical trait. So that's the end of mutations. I want to thank you for listening, and I hope you learned something new. Thank you.